Hello students, how are you all? So poll has been launched. Quickly answer me through poll. My screen is visible and my audio is clear. Okay, so in today's class, we are going to discuss transport in plants. So as you all know that transport in plants are of two types, the short distance transport and a long distance transport. If I say the short distance transport are those transport which are taken within the cell. Okay, those transport which are taking within the cells or those transport which requires the movement which require across the membrane or the movement which is taking place between the two cells. These transport are known as a short distance transport, and long distance transport are those transport which require vascular tissues like xylem and phloem, because we have to cover a very long distance. So short distance transport could be diffusion, facilitated diffusion, active transport. These are all of the short distance transport, and long distance when it involves the vascular tissue, that is xylem and phloem. In xylem, we are going to study the ascent of sap, and in phloem, we are going to study the mass flow hypothesis, which was proposed by Munch. So diffusion, as you know, this process does not require energy. So it is a passive process. Facilitated diffusion, it is also a passive process because it does not require energy. Active, it requires energy. It's an active process. In detail, we are going to do in the next one. Diffusion. Diffusion is when there is no requirement of energy. So no ATP. It's a very slow process. Active process is a very fast process. Remember this. Diffusion is a very slow process. And in diffusion, the movement of molecule occurs from high concentration to low concentration. Okay? Movement is occurring from high concentration to low concentration, and we also call it as downhill movement. We also call it as the movement is along the concentration gradient. Please remember these points. If I say downhill movement, that means the molecules are moving from high concentration to low concentration. If I'm saying along the concentration gradient, that means the molecules are moving from high concentration to low concentration. Remember these points. And diffusion is not dependent on living system. Important point. Remember this also. Movement, I told you, along the concentration gradient. No energy is required. That is why diffusion is known as a passive process. There are many factors which affect the rate of diffusion, like the gradient of concentration. Gradient always means difference. You can see this in the diagram. There is a difference in the concentration. At one point, the concentration is high. At one point, the concentration is low. So there's a gradient of concentration should be there for the diffusion. Permeability of the membrane. On the basis of permeability, the membrane could be a permeable membrane, impermeable membrane, selectively permeable membrane, or semi-permeable membrane. Temperature and pressure. These factors also affect the rate of diffusion. The rate of diffusion would be high if there's an increase in temperature. If there's an increase in pressure, the rate of diffusion will be high. Remember all these points. Second one says facilitated diffusion. I told you there's a term diffusion. That means it does not require energy. So here also no ATP is utilized. It's a passive process. Okay. But there's one important feature of facilitative diffusion, that is the presence of specific proteins. Facilitative diffusion requires protein for the movement of the molecule across the membrane. You can see this. This is outside of the cell. Lipid soluble. As you know that our membrane is phospholipid bilayer, right? Our membrane is phospholipid bilayer, right? So those molecules which are lipid soluble, they can easily cross the membrane, right? But those molecules which are polar in nature, what about those molecules which are hydrophilic in nature? These hydrophilic molecules, they require a special protein, and the movement of these molecules is occurring through the process is facilitative diffusion. And this process is highly specific because the proteins are highly specific. These proteins will able to bind only to a specific molecule. Okay, so there's a selective in nature. These molecules are these proteins are selective in nature. They show high specificity. And the porins, remember the porins, the membrane of mitochondria, plastid, chloroplast, and even the bacteria, they have porins. These are the large pores, so that the small molecule or the molecules which are smaller in size, they can easily move through these porins. There's a term is aquaporins, and 8 different aquaporins, they form a one water channel. And these water channels, they allow the movement of the in the same direction. Then it is symport. Okay, like sodium and glucose is an example of symporter. Antiport, when the two molecules are moving in opposite direction, this molecule A is moving in this direction, or molecule B is moving in this direction. They're moving in opposite direction, beta. Then it is antiport. The movement is antiport. Example like sodium and proton exchanger. Uniport, the molecules when they're moving in one direction, and the movement of that molecule is independent of other molecules. Then it is uniporter. Moving to next is active transport. I told you active means there's the utilization of ATP, and the movement of molecule is against the concentration gradient. Remember this. That means uphill movement. Remember, in diffusion and in facilitated diffusion, the movement is along the concentration gradient. From molecules are moving from high concentration to low concentration. The movement is along the gradient in case of diffusion and facilitated diffusion. But in active transport, the movement is against the concentration gradient. That is uphill transport. Movement of molecule is from low concentration to high concentration. That is against the gradient. It is an active transport. It requires energy. It requires special proteins. Yes. Active transport also requires the special protein, and these proteins are specific in nature. Examples of active transport is a sodium potassium pump. So active transport also requires specific proteins. Okay, and the movement is uphill against the concentration gradient. Remember this point, beta. Moving to next, this table, very, very important. It is given in NCERT. Please remember this table. It's very important. Number 1 says require special membrane proteins. I told you membrane proteins are required only in facilitated diffusion and active transport, but not in simple diffusion. Highly selective. Facilitated diffusion and active transport, they both require special proteins, right? So they are highly selective. Transport saturate. So when the proteins, like in facilitated diffusion and active transport, they require protein for the movement of molecules, right? And when they require the transport will be maximum when these proteins, they get saturated, all the proteins are used up, then the transport will be maximum. Okay, you observe this in facilitative diffusion and in active transport, uphill transport. When the movement of molecule is against the gradient, against the concentration gradient, you observe this in active transport. Okay, because in simple diffusion and in facilitative diffusion, the movement is downhill, that is along the concentration gradient, requires ATP energy. So ATP is required only in active transport. Okay, remember this point, beta, is very important. I hope this is clear, students. It's very important. Remember this point. Next is the water potential. Water potential is defined as the kinetic energy of the water molecule. If this is a beaker, okay, it's have a lot of water molecules. The kinetic energy of these water molecules will be high. There's a random motion and the motion will be very fast, okay? But if I start adding the solute to this to this solvent or the water, if I start adding the solute, then the solution will have a low water potential, and the magnitude by which the water potential is decreasing, the magnitude by which lowering the water potential, that is known as a solute potential. Because once I start adding the solute, the randomness, the motion of the water molecule get reduced. Okay. And the water potential of pure water is 0. Remember this. And solute potential is denoted by psi s, and it is always negative. Remember this. Pressure potential is denoted by psi p and is usually positive. Static pressure is positive, but the pressure which is created in the water column in the xylem vessel, right, because of the transpiration pull, that pressure is negative. And remember this equation. Very, very important. Remember this. This is a relation between the water potential, solute potential, and the pressure potential. Water potential of a cell is affected by both solute potential and the pressure potential. Very important. Moving to next is osmosis. As we all know, osmosis is the movement of water across a differentially or a selectively permeable membrane. Okay, this is a membrane, right? And the water, as this Thistle funnel contains sucrose solution. That means the solute concentration in the funnel is high, right? That means there is low water. So the water always moves from high concentration to low concentration. That is from high water potential to low water potential. So the water will start moving. End osmosis is taking place. And you can see this water, it is rising in the funnel, right? But if I apply the external pressure, if I apply the external pressure to prevent the entry of the water in the osmotically active solution, that pressure is known as osmotic pressure. Remember this point, beta. And more the solute, more will be the osmotic pressure. Okay? Because if okay, let me destroy. If this is a cell, Okay, it having a very high solute. Okay, if the solute is more, that means there will be less water, right? If the solute is more, then there will be less water, and the water will start moving from outside, because the water will move from high concentration towards the low concentration. So water will start moving. That means more pressure is required to stop the entry of the water, because more the solute, more will be the pressure to stop the entry of the water. So this pressure to prevent the entry of the water is known as the osmotic pressure. And rate of osmosis depends on both the pressure gradient and the concentration gradient. Yes, concentration gradient automatically develop, right? Because in this, there's a low solute. Sorry, in this, there's a high solute, right? And low water. Water is low. And in outside, the water is high. So the gradient of water concentration has developed, and the water will move from high concentration to the low concentration. Pressure gradient also developed. That is osmotic pressure, and water always move from low osmotic pressure to high osmotic pressure. Remember this. Next come is plasmolysis. Plasmolysis is a reversible process. Remember this, it's a reversible process. And there's a three type of solution: isotonic, hypertonic, and hypotonic. In hypotonic, the cell will have high solute and low water. So the water from outside will move in. So end osmosis will take place. And when the water moves in, the water start putting the pressure on the walls of the cell. So cell become turgid. In hypotonic, the cell become turgid. Whereas in hypertonic, the solutes are more in the external solution than in the, as compared to cell, the solutes are more in the external solution. That means there's a less solute in the cell, right? If there's a less solute, that means the water concentration would be high. So water will move out. So exosmosis will take place. And there's a shrinking of the protoplast of the cell takes place. And because of that shrinking, the cell become plasmolysed. Remember this. In isotonic, there's an equilibrium. The amount of water which is going out, the same amount of water is coming in. So there's a balance, there's an equilibrium in isotonic solution. Remember this points. Very important. Next is imbibition. You have observed that during the rainy season, the wooden doors they get swollen. It's very difficult to lock the door, right? In rainy season, we have observed this thing, right? And if we put the seeds in the water, then the seed gets swollen. There's an increase in the volume. There's an increase in the surface size of the seed. It's because of the imbibition. Imbibition is the diffusion when the water is absorbed by the solid colloids, causing them to increase in volume. You can see, first there's a dry seed, then the seeds, they have increase in size, they get swell. And the movement is along the concentration gradient. That is the imbibition. There's a downhill movement. Remember this. Moving next is the long distance transport. So long distance transport cannot be done by diffusion, because diffusion is a very slow process. No energy is required in diffusion. Okay, so the water and minerals and food, they are generally transported by the mass. Okay, and mass flow is the movement of substance in a bulk from one point to the another point because of the pressure differences between the 2 points. You can see in xylem unidirectional water will move from the roots to the different parts of the plant. In phloem, bidirectional movement occurs. Next movement is apoplastic movement and symplastic. Now it's clear, beta? Now you can hear my voice? Okay, okay. So there are two types of movement. One is apoplastic, another one is symplastic. Apoplastic movement, it takes place through the dead spaces of the cell. Cell wall. Cell wall is a dead cell. Remember, cell wall is dead. Second is intercellular spaces. And this movement is very fast. Apoplast is very fast. Why it is fast? Because there is no hindrance. Because the movement is taking through the dead spaces. Right? So there's no hindrance. Whereas in SYMPLAST, the movement is taking place through plasmodesmata bridges. That is, cytoplasmic bridges that are plasmodesmata. Okay, and the SYMPLAST movement is slow because there are hindrance, right? Once in the cytoplasm, there's a lot of things. There's many cell organelles, so the movement sometimes get interrupted or there's a hindrance. You can see this apoplastic movement. The movement is taking place through the intercellular spaces. But when the water reaches the endodermis of the root, the movement becomes symplastic. Why? Because there's a presence of Casparian strips. And these Casparian strips, they are highly suberized. And because of the deposition of the suberin, the water, they cannot move in. These walls, because of the presence of the Casparian strip, the walls become impermeable. So the movement of the water gets stopped. So then the movement occurs through SYMPLAST. And once the water reaches the xylem, the movement of water again become apoplast, because xylem consists of vessels, tracheids, and these are dead elements. And I told you the movement through the dead element, through the cell wall and intercellular spaces is apoplastic. Remember this. Moving next is the root pressure. So root pressure, it is a positive pressure. And we observe root pressure during early morning and night. Why? Because the rate of absorption is high and the rate of transpiration is low. Okay, and this pressure is responsible for pushing up the water to small heights. So we generally observe root pressure in herbaceous plants because root pressure can push the water only up to a small height. For longer height, we need transpirational pull that we are going to discuss in a minute. And very important is guttation. Guttation is also due to root pressure. You can see these liquid droplets. It's because of the guttation. The loss of the water in the liquid form. Remember, I'm giving emphasis on this. When the loss of the water is taking place in the form of liquid, water is losing from the leaves or the parts of the plant in the form of liquid, that is guttation. But if the water loss is in the form of water vapor, that is transpiration. Remember this point. Transpiration is taking place through stomata, right? But in guttation, there's a pores known as hydrothodes. And these pores are permanently open. And the water, in case of guttation, is impure because it also contains the solute, but the water but the loss of the water in the form of water vapors in transpiration, that is pure because it does not contain any solute. Remember this point, very important. Moving next is the ascent of sap, transpirational pull. Okay, so when the light is falling on this leaf, the water is evaporating in the form of water vapor, and the loss of water in the form of water vapor, we call it as transpiration, because continuously water is evaporating from the leaf. So a maximum amount of water is absorbed through the root. So maximum amount of water absorption by the root is taking place through transpiration. Remember this point. And there are some properties of transpiration pull. Number one is the cohesion. When there's an attraction between the water molecule, you can see this. There's this one water molecule, and there's a bond between them. So cohesion, hydrogen bonding. So the cohesion is the attraction between the two water molecules, right? Between the same molecule, that is cohesion. If there's an attraction between the different molecules, then they are adhesion, like the attraction between the water molecules and the polar surfaces of the cell wall. So it is adhesion. Third is surface tension. And all these properties, they contribute to the high tensile strength and capillarity. Capillarity is the rise in the tube. Okay, these are very important properties. Remember this. Next one is function of the transpiration. First, it creates a transpiration pull for absorption. Because if the transpiration pull is created, then the more amount of water can be absorbed through the roots, and these water, they can move to a tall heights. They can move to a very long, they can cover a very long distance in the tall plants. Supply of water, yes, for photosynthesis. Water is very important for photosynthesis, right? Transport of mineral from soil to all the parts of the plants. It also taking place through transpiration. Transpiration play a very important role in cooling. It decreases the temperature by 10 to 15 degrees Celsius, cool the surface area of the leaf, and maintain the shape and structure of the cell by turgidity, by keeping the cell in turgid. You can see this structure of stomata. The inner walls of the guard cells are thick and elastic. Why it is elastic? Because the stomata, they continuously opening and closing of the stomata is taking place. It has to be elastic nature, right? If it is inelastic, then it will break, right? So when something is opening and closing, it has to be elastic, flexible. So the inner walls of the guard cells, they are elastic in nature. They are thick and elastic in nature. The outer walls are thin. Remember this point. And the chloroplast is only present in the guard cell of the stomata. Remember this point. Moving next is the uptake of mineral ions. The uptake of mineral ions majorly is active process. Remember this. Active absorption of mineral ions occur mostly, but sometimes it is passive as well. When root absorbs the water from the soil, some minerals, they passively moves along the water into the root. But the concentration of minerals in the cytoplasm of the root is more than the soil. So soil has low concentration of minerals, and the root cytoplasm has high concentration of mineral. So mineral is, and the absorption of mineral is taking place from low concentration to high concentration. That is against the gradient. So we require energy. That is why it is an active process. Remember this point. Very important. It is already written here. The concentration of mineral iron in the soil is usually lower than the concentration of minerals in the root. That is why the process is active, because we are moving against the gradient. But some ions also move into the epidermal cells passively. Remember this. So ion absorption from the soils are both passive and active process, but majorly active. If the question says majorly the mineral ions are absorbed, dash, it's active or passive, then you have to answer active, because they are asking about majorly or the majority. Translocation of ions. So this one is an older leaf. Okay. These are the older leaves, okay, and these are young leaves. So when the older leaves are about to die, they are in the senescent stage and they're about to die or about to fall off. In that case, all the mineral ions or the elements which are mobile in nature, like phosphorus, sulfur, nitrogen, and potassium, they start moving towards the younger leaves. But there are some elements which are immobile, like calcium. Calcium is immobile. Very important. Remember this point, beta, that calcium is immobile because a part of a structural component, so it cannot move out. Okay, it's clear now. Translocation of mineral. Now moving next. Mass flow hypothesis. It is proposed by Ernst Munch. So in mass flow hypothesis, whatever, this is a leaf. And you all know leaves are the source. They act as a source because their photosynthesis is taking place. Okay, so the two processes are taking place, loading and here unloading. So loading takes place at the source and unloading takes place at the sink. And both the process, loading and unloading, are the active process. Remember this point. Both are active process. Both require energy. So when the loading occurs in the sieve tube cell, the concentration of the solute in the sieve tube cells increases, right? Then the water potential goes down, because if the solute is increases, the water is less, right? So the water from the adjacent cells, they start moving into the sieve tubes and osmosis is taking place. Okay, so there's a water gradient, water potential gradient has been established at the site of loading. And when the water moves in and osmosis takes place, the cell increase in size, they put the pressure on the wall. So there's a high turgor pressure. At the loading stage, there's a high turgor pressure. And when this solute moves in the phloem towards the sink. Sinks are the site where the turgor pressure is low. So the sugar solution flows to the region where there's low turgor pressure. Okay. Once it reaches to the sink, the unloading takes place. That means all the solute are moving out of the sieve tubes. When all the solute will move out, what will happen? Again, the water potential gradient will establish, right? Because all the solute will move out. Now the cell will have a high water potential. Then if having a high water potential, then the water will move from high water potential to low water potential, right? So the water will move out of the cell. Exosmosis will take place. Remember these points. It's written here, the loading of the phloem set up the water potential gradient and that facilitates the mass movement in the phloem. At the sink, incoming sugar are actively transported, actively. Actively, very important. Remember this, both loading and unloading are active process. It's transported out of the xylem and moved as a complex carbohydrate. Loss of solute produce a high water potential. I told you at the sink, when the unloading takes place, it leads to a high water potential in the cells and the water will move out and returning eventually to xylem. Girdling experiment. It was proposed by Malfigi. Okay. And what happened? They removed the bark. A ring of bark has been removed. And bark is consist of all the tissues outside the vascular cambium. So it consists of periderm plus your secondary phloem. Remember this point. What comes under bark is the periderm plus the secondary phloem. All the tissues outside the vascular cambium are known as bark. So when the scientist, what he did, he removed the bark. So that means he removed the periderm, he removed the phloem, right? And you know that food which is synthesized in the leaf, that is source. So food always move from source to sink. Movement of food is from source to sink, right? So food will move from leaves to root. But at this point, there's an interruption because the bark has been removed, the secondary phloem has been removed. So what he observed the swelling. The ring becomes swollen at the end because the food get deposited here. So there's a swelling of the ring. So what he observed from this experiment, that phloem is the tissue which is responsible for translocation of the food. Remember this point. I hope everything is clear. Now we are moving with some questions. The previous year question. First question said addition of more solute in a given solution will. Option 1 says lower its water potential. Option 2 says makes it water potential 0. Option 3 says not affect the water potential at all. And option 4 says raise its water potential. So what do you think? What will be the correct option for this question? Okay, Chichuni says 1. Okay. If you want me to cover more questions, please reply quickly. Okairu Gabo says 1. I hope I'm pronouncing your name correctly. MS Gauri says A. Okay. Very good, everybody. All of you have answered it correctly. Addition of more solute in a given solution, it lowered its water potential. It's very important. When the pure water has a high water potential and that is 0, but the moment you start adding the solute to that what solution, when you start adding the solute, what will happen? The random motion of the water molecule gets reduced, so it's lower its water potential. That is the kinetic energy of and water potential is generally defined as the kinetic energy of the water molecule. So it lowers the water potential. Moving next. Which of the following statement about facilitated diffusion is incorrect? Incorrect, they're asking. Number 1, porins are involved in this process. Number 2 is movement of molecules occur against the concentration gradient. Number 3 says ATP is not required for this process. And option 4 says the special proteins of the membrane help in this process. Quickly answer, everybody. They're asking about the incorrect statement about the facilitative diffusion. Okay, Chinchuni says 2. Vijay, Ritu, Etvi, what do you think? Rahul, Vijay, Pankaj. Okay. So question says, which of the following statement is incorrect about facilitative diffusion? They're asking about the incorrect statement. Number one says porins are involved in this process. Yes. Porins are present in the outer membrane of mitochondria, chloroplast, and the bacteria. And these porins, they are larger in size, so the smaller molecule, they can easily pass through it. Right? That is a correct statement. Number two says movement of molecule occurs against the concentration gradient. Beta movement of molecule in diffusion as well as in facilitative diffusion is along the concentration gradient. That is from high concentration to low concentration. We also call it as downhill movement. Against the concentration gradient is a part of an active process, not the facilitative diffusion. Remember this point. And they're asking about the incorrect statement. So incorrect statement is number two. So correct option is number two. The movement of molecule occurs against the concentration gradient. In facilitated diffusion, it is along the concentration gradient. ATP is not required. True. ATP does not require in diffusion or in facilitative diffusion. It is required only in active, right? Special protein of the membrane help in this process. Yes, facilitative diffusion do involves the special proteins. Moving to next. Two cells A and B are contiguous. The cell has an osmotic potential, 10 atmosphere, turgor pressure. They're asking about the movement of the water. Okay. Option 1 says movement of water from cell B to A. Option 2 says no movement of water. Option 3 says equilibrium between the two. Option 4 says movement of water from cell A to B. Okay, beta. Beta, facilitated diffusion. We know that there is a two diffusion. One is simple diffusion and another one is a facilitated diffusion, right? In simple diffusion, what happens? The molecules which are lipid soluble, they can easily cross the membrane. They does not require any special protein. But what about the hydrophilic molecules or the polar molecules? Because membrane is phospholipid bilayer, right? These molecules cannot easily pass the membrane, right? So the movement of hydrophilic molecules or the polar molecule, they require special proteins. Okay. If the movement of the molecule is facilitated by the specific protein and that movement is along the concentration gradient, that movement is passive, that does not require any energy, that movement is known as facilitated diffusion. I hope it is clear now. Contiguous means they are adjacent. बहुत पास है, very close to each other. Kai, Rayu, Gabo, I hope it is clear now. Okay. Now moving to the next question. This is very important. Do people know about DPD? Let me ask you this. Diffusion pressure deficit. Quickly answer me in the chat box. And if you know about DPD, then quickly answer what you think, what will be the correct option for this question. Okay, okay. Let me tell you this. So beta, DPD is diffusion pressure deficit. We also, in local term, we call it a cell thirst. When you feel thirsty, you drink water, right? So I'm going to explain the simplest way that DPD is a diffusion pressure deficit, and we also call it as suction pressure. We also call it as cell thirst. For example, this is a cell A. Let's keep it as 1 because I don't want you to get confused with the question. And this is cell 2. Okay. This cell has a lot of solute, high solute. So tell me, if the cell having a high solute, what will be the concentration of water? What will be the concentration of water if the cell is having a high solute? Low. Very good. If the cell is having a high solute, that means the cell will have a low water potential, right? So cell thrust will be more or less. This cell will be thirsty or not? Because it's having a very low water. This cell is having a high solute. That means low water potential. So cell's thrust will be more. So this will be having a high DPD. Okay, because the cells require more water because it's having a very high solute. So cell thrust will be more. Sorry for the interruption, students. So next is cell number 2. So cell number 2 is having a very low solute. If the solute is low, that means the water will be high. More water. So if the cell is already having a water, so cell thirst will be low, right? So in this case, DPD will be low. And water moves from, can anybody tell me, water moves from high concentration to low concentration, right? Is it clear to everyone that water always moves from high concentration to low concentration? So here the water will move from this because this cell is having a low DPD, it's having a high water potential. So water will move from cell 2 to cell 1. Okay? Now you can easily answer this question. The question says that two cells A and B are contiguous. That means they're very adjacent. Cell A has an osmotic pressure 10 atmosphere, turgor pressure 7, diffusion deficit 3 atm. DPD beta is also OP minus TP. This is a formula of DPD. Okay, remember this. But I'll tell you a trick. There's no need to, you don't have to look at even the atmospheric pressure, the turgor pressure. Just look at the DPD. Because just I told you, the water is from the low DPD is to high DPD. Movement of water is from low DPD to high DPD. So cell A is having a 3. DPD is 3 atm. Cell A, DPD, 3 atmospheric pressure, 3. Cell B, DPD is 5. So water will always move from low DPD to high DPD. Water will move from cell A to cell B. So option 4 is the correct answer. Movement of water from cell A to cell B. Okay. This is how you can save your time during the exam. Try to apply these tricks. Very good, MS Gauri is 4. Now moving next. The ascent of xylem sap in plants is mainly accomplished by the option 1 says size of the stomatal aperture. Number 2 says distribution of stomata on the upper and the lower epidermis. Number 3 says cohesion and adhesion between the water molecules. And number 4 says root pressure. What do you think? What will be the correct option for this question? The ascent of xylem sap in plants is mainly accomplished by the okay. Try to remember the properties of transpiration. There were 3: cohesion, adhesion, surface tension, and all these properties they contribute to high tensile strength. Okay? And high capillarity, because of which the water can travel to a long distance. Accomplished is beta achieved. It's an English term. It's not a scientific term. So size of the stomatal aperture, no. Distribution of stomata on the upper and lower epidermis. Option 3 says cohesion and adhesion. Yes. Correct option is 3. Option 4 says root pressure. No, beta. Ascent of xylem sap is mainly accomplished by the cohesion and adhesion between the water molecules. Very good, Chichuni. You answered it correctly. Moving to next is when a carrier protein facilitates the movement of two molecules across the membrane in same direction, it is called. It's a very easy question, beta. When a carrier protein facilitates the movement of the molecules of movement of two molecules across the membrane in the same direction, they're talking about the same direction. What would you think? What will be the correct option? Number 1 says uniport, option 2 says transport, option 3 says antiport, and option 4 says symport. Chichuni says fourth. Okay. Ritu, Hetvi. What do you think? What will be the correct answer for this question? So there's symport, there's antiport, and there's uniport. Movement of two molecules in same direction. Symport. Movement of two molecules in opposite direction. Antiport. And the movement of molecule in one direction, independent of the other molecule, then it is uniport. So the correct answer for this question is symport. That is fourth. When a carrier protein facilitates the movement of the two molecules across the membrane in the same direction, they're asking about the same direction. And same direction is symport. So correct answer will be fourth, that is symport. Clear to everybody? Chichuni, very good, beta. Next is selects the incorrect statement. Some statements are given. You have to select which statement is incorrect. Option 1 says transport of molecule in phloem can be bidirectional. Option 2 says movement of mineral in xylem is unidirectional. Option 3 says loading, unloading of sucrose at sink does not involve the utilization of ATP. Option 4 says element most easily mobilized in plants from one region to another are phosphorus, sulfur, nitrogen, and potassium. What do you think? What will be the correct option for this question? I told you when I was teaching you mass flow hypothesis. Okay. So I'll explain this quickly because we are running out of time. Option 1 says transport of molecule in phloem can be bidirectional. Yes. In phloem, the transport is bidirectional. Okay. But in xylem, the transport is unidirectional. Remember this, phloem bidirectional, xylem unidirectional. Okay, option 1 says transport of molecule in phloem can be bidirectional. Yes. Option 2 says movement of mineral in xylem is unidirectional. Yes, because the water is moving unidirectional. So the minerals, all the minerals which are moving along the water in xylem are also moving in the unidirectional. Number 3 says unloading of sucrose at sink does not involve the utilization of ATP. Beta, unloading and loading both are active process. That means they both require ATP. But this option says that unloading of sucrose at sink does not involve the utilization of ATP. So this is the incorrect statement. And correct option with respect to the question, because the question is asking about the incorrect statement. Option 4 says the element most easily mobilized in plants from one region to another are phosphorus, sulfur, nitrogen, and potassium. All these elements are mobile elements, but the calcium is immobile element. Remember this. I hope it is clear now.